The trachea is anterior to the esophagus, and please never, ever, ever call it the windpipe going forward. A windpipe, calling it the windpipe makes you sound like you are uneducated, and we are anatomists and healthcare professionals, so use the term trachea. It is going to extend from the larynx to the level of approximately T5. The wall will have 3 layers, and those layers look a whole heck of a lot like layers of vessels. What a surprise. The deepest layer is going to be formed of ciliated pseudostratified epithelium, which contains goblet cells. That is going to be the mucosa. Deep to that will be the submucosa with connective tissue, which will contain what are known as seromucus glands. What a surprise, more mucus. And the deep, most superficial layer is going to be the adventitia, which is going to be composed of connective tissue. That will encase those C-shaped rings of hyaline cartilage. Why is it crucial that the larynx and the trachea are formed, and the primary bronchi, actually going down to secondary and some of the tertiary, or some parts of the tertiary bronchi, are formed of hyaline cartilage? Why is the fact that there's so much hyaline cartilage in the respiratory tract important? Think about that, and we will address it. The trachealis muscle is going to connect the tips of the C rings posteriorly, and it will contract during coughing to help expel mucus. It's going to be another function, and we'll talk about that momentarily. The carina is a little spar of cartilage on the very last of the tracheal cartilage rings, and it is the point where the trachea is going to branch into the 2 main bronchi. So the structure of the trachea will be up to about 20 incomplete rings, those C rings of hyaline cartilage, and the trachealis muscle will interconnect the tips of those C rings. It will be lined by mucous membrane. We talked about how it's going to be lined by pseudostratified columnar epithelium. And then we already talked about the carina. The reason that hyaline cartilage in the larynx, in the trachea, in the bronchi, so important because the hyaline cartilage gives enough structure that the trachea will not collapse. It keeps it open, even under some fairly severe pressure. You don't want that to collapse, because if it does, you're dead. Ton of little videos here. Please watch them. They give you excellent information. The dissection videos are particularly interesting, but warning, they are dissection of sheep, so be prepared. So let's take a look at this. So you can see the trachea, you can see the C rings, and you can see the trachealis muscle, which is going to be here. And here's our friend, the esophagus. So one of the reasons that it's important that we have the trachealis muscle that connects those C rings is this. If you take a super big bite of an apple or food or whatever, and you have this giant bolus traveling down your esophagus. Hello, go back that way. The trachealis muscle can actually bow inward to allow that bolus, that food that you've eaten and are swallowing, down through the esophagus without damaging the trachea in any way, shape, or form. So not only does the trachealis muscle help you cough and expel mucus, it also allows you to swallow successfully if you have a particularly huge bolus that you're trying to get down into your stomach. This should look totally familiar because we have seen the slides like this. If you were with me for the histology portion, and even if you weren't, you should say, oh yeah, right, I get it, seen them. Please note the hyaline cartilage. Hyaline cartilage has those lacunae that have the chondrocytes in them, and the matrix is going to look nice and smooth and glossy. This is going to be the pseudostratified columnar epithelium, and there's the cilia, and here are the pseudostratified columnar cells. What fun. And I love this because it shows you an electron micrograph of cilia. Just beautiful. Now, we're down at the bronchi. We're going to talk about the bronchi and the subdivisions. There are, so literally it's called bronchial tree for a reason, because there are going to be a significant number of branches off the bronchi. Okay, so they're going to be 23 orders of smaller and smaller and smaller and smaller and smaller and smaller branches, which form that respiratory or bronchial tree. So the tips of the bronchial tree to the conducting zone structures, down to the respiratory zone structures. And we're going to be talking more about each of those. We'll talk about the respiratory zone structures as we get into the histology more. Now, primary bronchus is going to be one into each lung, so there'll be one on the right and one on the left. The one on the right primary bronchus is shorter, wider, and more vertical than the one on the left. The primary bronchus are going to branch into secondary bronchi, tertiary bronchi, and they will divide into smaller and smaller and smaller branches, which are bronchioles, and smaller and smaller and smaller bronchioles into terminal bronchioles before you get to the respiratory bronchioles. So for the secondary bronchi, they're going to be 3 secondary bronchi on the right and 2 on the left, because the lungs have different numbers of lobes. And then again, they'll branch into smaller and smaller and smaller divisions of that. Now, it shows you what's going on. There is the trachea. You can see the primary bronchi on each side. You can see the secondary bronchi on each side. And you can also see very nicely a cross-section, transverse section of the trachea and the esophagus posterior to that. Which you've already seen, but there it is again. Now, the terminal bronchioles are going to continue to branch down into the respiratory bronchioles, into the alveolar ducts, into the alveolar sacs, which are going to contain clusters of alveoli. They look like clusters of grapes, and then the alveoli. And we're going to talk more about these shortly, and we'll talk about the alveoli again when we get to the histology part. So this is a cool picture because if you take a look at the surface area of a tennis court, it's about 195 square meters. And the surface area of the alveoli is, if you flatten them all out, it's about 180 square meters. So the next time you're playing tennis, think, holy cow, this is about the size of the surface area of the gas exchange membranes in my lungs. I love that. Just fascinating to me. This shows you the respiratory tree in the lungs. You can see that there's extensive branching that continues on. And again, I've already mentioned this and we'll mention it again. Each lung has a different number of lobes. I wonder why. Think about why that might be. Look at the lung on the left. There's that weird little notch thing there. This guy right here. Wonder what that's for. You probably already know. Now, with the extensive branching, this is where we start to talk about what happens. There are going to be epithelial changes from ciliated pseudostratified columnar epithelium to non-ciliated columnar epithelium in the terminal bronchioles, then down to simple cuboidal epithelium, and then, once you're in the alveoli, simple squamous epithelium. The cartilage will decrease from rings in the trachea and the primary bronchi into irregular plates, and then once you hit the bronchioles, you will have elastic fibers that are going to replace those cartilage. What do you think the function of those elastic fibers are? You got it, because those bronchioles are going to need to expand and contract, and those elastic fibers allow for that. As we move down or up, down the bronchial tree, smooth muscle will increase. It will be innervated both by the autonomic nervous system and by hormones. How? Well, the PNS and histamine will constrict the bronchioles, so you'll have bronchoconstriction. Remember those elastic fibers we just mentioned? There you have it. And the sympathetic nervous system and epinephrine will dilate bronchioles, bronchodilation. And I got this wrong. This will be the parasympathetic nervous system and histamine. My mistake, I'm sorry. So parasympathetic nervous system and histamine constrict the bronchioles, and cause bronchoconstriction. For bronchodilation, you have sympathetic nervous system stimulation, and epinephrine are going to trigger that. Now, from the bronchi through the bronchioles, you're going to have structural changes, which we have already talked about, but this sums it up. We know that cartilage rings become irregular plates, and we know that at the level of the bronchioles, you will have elastic fibers and not cartilage. We know that there are going to be significant epithelial changes from pseudostratified columnar epithelium, which is ciliated, down to simple cuboidal epithelium, and cilia and goblet cells will become sparse. Once you get down to the respiratory membranes, you're talking about simple squamous epithelium. And we also know that the relative amount of smooth muscle will increase, which will allow for constriction of the bronchioles. Now, the lungs themselves, they are paired and cone-shaped organs. Think way back to when we talked about membranes, and we talked about the difference between mucous membranes and serous membranes. The lungs are going to be covered by serous membranes, which are called the pleural membranes. All serous membranes have two layers, the visceral layer and the parietal layer. The visceral layer is going to sit right atop the organ and follow its contours. The parietal layer will be the more superficial layer. In between those layers will be the serous cavity, and in this case, the pleural cavity, which is going to contain serous fluid. Pleural fluid has a very, very high surface tension. And these serous membranes are unlike the peritoneal serous membranes and the serous membranes that you find. Where else do you find them? Who remembers? The heart. The pleural fluid does not mean that you need to have lubrication. It is going to allow the pleural membranes to act as one. And we'll talk more about this soon. It shows you the pleural membranes. So the visceral pleura is a deeper layer. It is going to literally cover the contours of the organ. The parietal pleura is going to line the thoracic cavity, so it's going to be attached to the thoracic cavity. Remember, that's going to be very important when we're talking about inspiration and expiration. And that virtual space in between the pleural membranes is the pleural cavity. Gross anatomy of the lungs. We already talked about this. Each lung, some of this, each lung is going to be divided into lobes by fissures. The right lung has 3 lobes. The left lung has 2. We know that each lobe is going to receive a primary bronchus and then a secondary bronchus, and those bronchus will continue to branch. Each lobe will also be divided into lobules, and each lobule will be wrapped in elastic connective tissue. It will contain a lymphatic vessel, a blood vessel, a venule, to change the spelling of that, and a branch of the terminal bronchiole. The apex is the tip that is superiorly positioned. It's deep to the clavicle, so if you palpate your clavicle, yes, that is how high the apex is. in your thoracic cavity. The base is going to be the inferior surface that will rest upon the diaphragm. The hilum is the site where all of the blood vessels and the bronchi and lymphatic vessels and nerves are going to enter and exit. The left lung is going to be smaller than the right, and that is because it has to make room for the heart. So that notch that I pointed out, the left lung, is known as the cardiac notch because you need to have room for the heart. The left lung will be separated into superior and inferior lobes by the oblique fissure. Oops, went backwards, sorry. Now, it shows you a diagram, shows you the cardiac notch, shows you the different lobes in each of the lungs, and the diagram on the right shows you very nicely the hilum and all the vessels and area for the nerves, and also points out the apex and the base. This shows you the heart relative to the lungs on the left, and if you remove the heart, you can see what is known as the root of the lung, which is where the vessels and airways enter and exit. Fun fact. So each lung is going to contain approximately 300 million alveoli. When the structures are filled with air, the lungs can actually float. And this is going to be important, at least historically, because way back when, medical examiners used to use what was known as the lung float test during autopsies to determine if a baby was stillborn. If the lungs floated, the baby was born alive. If the lungs didn't float, the baby was a stillborn baby. And this is pretty accurate 98% of the time, according to a 2013 study. Pretty amazing. Now, I'm going to stop here because we will talk about the microscopic anatomy in a separate section.